อ Hi, everyone. Thank you for tuning into our third seminar of our ABL Seminar Series 2022. So today we have Dr. Eileen Lacey as our speaker. And before we begin, just a reminder that you can actually ask questions in the YouTube chat at any time, and then we'll relay the questions to her at the end during the question and answer period. So a little bit about Dr. Lacey. She is the departmental co-chair, professor in the Department of Integrative Biology, as well as a curator in the Museum of Vertebrate Zoology at the University of California, Berkeley. So Dr. Lacey completed her BSc at Cornell University, which was followed by her master's and PhD at the University of Michigan. And during this time, she focused on reproductive strategies of male ground squirrels. And so her current lab at Berkeley investigates behavioral ecology as well as social behavior of mammals with an emphasis on subterranean rodents. And so today she'll be giving a talk entitled Digging In, What Are Tuco-tucos Revealing About Subterranean Sociality? And so Dr. Lacey, you can go ahead. Thank you for joining us.
Thanks, Amanda, and welcome everybody. And even though it seems like we've been living our lives on Zoom for a couple of years, it still amazes me that I can be sitting here in my office in Berkeley and sharing my work with people throughout North America, the U.S., sounds like Europe as well. So welcome, and good morning or good afternoon, depending on what time zone you're in. So what I'd like to do today is share with you some of the work that I've been engaged in during my time as an active researcher in behavioral biology, particularly over the last decade or so, both the empirical work that I've been engaged in, and some of the thinking that has gone along with those studies. And along the way, I hope to introduce you to some really interesting animals that I've been working with, but that aren't really well known yet. So hopefully you can pick up some interesting science, generate some exciting questions, and get to know some pretty interesting little animals. To set the stage a little bit, I'm fascinated by animals that live in groups. I mean, looking at this picture, this collage of different group living species, it's hard to not get excited. Why are these animals living this way? Why do groups differ so much in structure? If you think about it, okay, all of the species shown here are group living, but the groups they live in vary in all kinds of ways, including group size, the sex, age, and kin structure of those groups, and in some cases, the breeding structure of the groups, meaning what proportion of adults of each sex actually produce offspring. And again, I look at pictures like this, and I'm just fascinated. I want to understand why those differences exist. As a result, much of my research has been driven by the following two questions. One, why do groups form? Or another way to phrase that, why do some species live in groups, but others don't? And the second question, why does group structure vary so much? And again, today I'll be sharing some of my efforts to try and pick away at these questions. These are big questions, so I don't pretend to expect that I will answer them fully. but maybe even start to generate a few insights for at least some lineages of mammals that I've been working with. So, taxonomically, as Amanda mentioned, my interest is focused on subterranean rodents. The term subterranean is an ecological designation that refers to rodents that spend their entire lives in underground burrows. Now, although the exact extent to which animals venture above onto the surface varies across taxa, the underlying idea is that subterranean species spend the majority of their lives and perform most critical life activities underground. Using that designation, turns out subterranean rodents, the animals designated as subterranean, provide a pretty nice comparative study system for several reasons. First, they're geographically nearly cosmopolitan in distribution, as you can see in the map in the upper left-hand corner of this slide. If we consider all rodent lineages that are typically designated subterranean, they occur in all major land masses except Australia and Antarctica. They also represent multiple phylogenetic origins for life underground, providing nice opportunities for studies of convergence and divergence. And at the same time, life in underground burrows is thought to be associated with a suite of selective factors that are common to all of these rodents, and therefore have resulted in a number of convergences in, for example, morphology, as well as a suite of shared life history traits. So again, just setting up a very nice comparative framework for asking questions about behavior and other phenotypic traits. So I'll pause here for a second, a little mammalogical quiz. Of the animals shown here, those photos, which one is the tuco-tuco? Before you can all Google it to get the answer, I'll go ahead and tell you that's the tuco-tuco down in the lower right. You'll see a lot more, hear a lot more about these animals today. So, to get started, I want to give you a little bit of my background, the backstory to how I got interested in this line of research, because it really does set the stage conceptually and empirically for what I'm doing today. For me, it all began with naked mole rats. Amanda and I were discussing these animals before the seminar began. They are just rude, but they're also super interesting in a variety of ways. And as an undergraduate at Cornell back in the Pleistocene, I almost literally stumbled into the lab of Paul Sherman at Cornell, who at that time was working with some of the first captive naked mole rat colonies that had been imported to the U.S. This was heaven, and I spent much of the next 4 years in hot, dark rooms watching naked mole rats carry bits of food around, build nests, dig through various obstructions that we created for them in their tunnels, trying to characterize the structure of the captive colonies that we had at Cornell. The result was one of the first descriptions of the division of labor in naked mole rat colonies. Now, we already knew there was a reproductive division of labor, meaning that within a colony there was only one breeding female and one to a few breeding males. But what I was involved in was really then characterizing what all the other non-breeding individuals in the colony do. It turns out they have pretty specific tasks that are related to body weight and to some degree to age. So some smaller non-breeders specialize on things like gathering food, building nests. Larger non-breeders tend to be more engaged in digging tunnels and defending the colony when threatened. And that was a lot of the early work that I was involved in. Collectively, these efforts contributed to our understanding, now well understood, that naked mole rats are group living, they engage in alloparental care, and they're singular breeding, again referring to that restriction of direct reproduction to only a single female. All of this led to the supposition or the statement, the hypothesis, that naked mole rats are a eusocial mammal. Historically, those same three criteria, group living, alloparental care, and singular breeding, have been restricted to describing highly social insects such as honeybees, ants, some wasps, and termites. This was the first suggestion that there might be a eusocial mammal. This was important. It wasn't just one of those gee whiz, this is cool moments. It actually had quite an impact on our conceptual understanding social behavior. As I mentioned, historically, the term eusocial had been restricted or applied only to highly social insects. And so I've tried to characterize this historical scheme here using some poorly defined axis of social complexity that includes somehow relationships between solitary species, plural breeding species, groups in which everybody's reproducing, and singular breeding species. But then historically, this divide between all of those taxa and eusociality, again restricted to insects. Well, the idea that naked mole rats might be social led to the proposal of an alternative scheme that's become known as the continuum view, depicted here, basically arguing that there is no evolutionary or selectively important break between everything and eusociality, but instead it's the same suite of selective factors just acting in different degrees and slightly different combinations that leads to any and all of these social systems. Now, that view has not been universally accepted, and it's led to, let's say, some lively discussion among the folks who care about these things. But again, I think the important point going back to the mole rats was they at least helped spark the discussion and have caused a lot of behavioral biologists to begin to rethink the comparative relationships between different forms of social structure. Now, to come back to the mole rats then, or this is kind of where I got to in terms of my undergraduate career, that mole rats were having a real impact on how we understood broad questions about social behavior. But at the same time, the more I thought about it, the more I thought, no matter how weird mole rats are, and this is before we knew they lived forever, and they seem to be immune to cancer and all the things that are going on now, we knew they were behaviorally extreme, but at the same time, really, it all came back to the fact that they lived in groups, that presumably none of those elaborate differences in behavioral specialization, division of labor, none of those things would evolve if the animals didn't live in groups. So I became increasingly interested in asking, what is it that leads to group living, particularly among subterranean rodents? And again, I wanted to restrict my search to subterranean species due to that shared suite of life history, ecological, and other features that we thought would favor convergence. It just limited the scope a little bit, and I thought might add some traction to my ability to make progress in understanding why these animals live in groups. Well, at the time I was asking this question, there weren't a lot of options known. We knew that there were social African mole rats, the naked mole rat, and some of its other relatives, but because I really wanted to work with something that was evolutionarily independent, I set the mole rats aside. That left me with a couple options, meaning the two other known social subterranean species. One is the caruru, a species that occurs in central northern Chile in the family Octodontidae. The other, at the time, was a recently described species of tuco-tuco, the colonial tuco-tuco from southern Argentina. For a variety of reasons, including logistic ideas, as well as the potential for within lineage comparisons, I opted to study the tuco-tucos. Well, this begs the question, these animals are not well known. What is a tuco? Answers vary. For those of us of a certain age, we may tend to think of Eli Wallach in The Good, the Bad, and the Ugly. Most of my younger colleagues are more of the Breaking Bad generation and often tend to think of Tuco Salamanca, the character in that series. Turns out that where I work in Argentina, tuco is also the name of a common pasta sauce. But then, of course, really what I'm thinking about is the rodent shown in the lower right. So, tuco-tuco, we are talking about a rodent. You'll learn more about them in a few moments. A little more formally, to give you a little background on the biology of tuco-tucos, they're members of the rodent family Ctenomyidae. The family contains a single genus, the genus Ctenomyidae, but it's a very speciose genus. We're currently at about 65 described species, and the numbers keep increasing. Again, these animals in general are poorly studied, and thus as people are going out and doing more field work, the taxonomy is being resolved, and it's generally resulting in an ever-increasing number of species of tuco-tucos. Despite the number of species involved, all tuco-tucos are subterranean. They all live in burrows. They all spend significant portions of their time underground. But again, presumably due to a shared suite of selective factors as well as shared evolutionary history, they're relatively conserved morphologically. The joke looking at these photos here is if you've seen one tuco, you've seen them all. And it's not too far off unless you're a real aficionado. So, just give you a little bit of an idea of the background of the biology, I should also mention the geographic distribution of the family is shown in the map here. It is South American, from the southern end of the Amazon basin down to Tierra del Fuego. They don't, however, cross the Andes very much into Chile, so it really is their distribution is east to the Andes. Well, with that little bit of background, I'll step into part 2 of the talk for today, and that is I'll tell you a little bit about the social structure of the colonial tuco-tuco, the species that I began working with. And the reason I started with this species was, again, very anecdotal information that these animals were doing something different and might be social. That's as much as I knew when I started out. It was a one-line description in the species description for colonial tuco-tucos, suggesting that there was something going on. To give you a little bit of information about the colonial tuco-tuco now, Ctenomyidae sociality, well named, it turns out. This species is currently endemic to a very small region of southern Argentina. The entire known geographic distribution is about 1,500 square kilometers, all contained within Parque Nacional Nahuatl, Argentina's flagship national park. I first visited this area in December of 1991, and based on what I saw, I established a long-term study site, the black dot in the map in the center of the figure, from which I've been working ever since. Just to orient you a little bit, you can see roughly where this area is in southern Argentina. Good landmark is the city of San Carlos de Bariloche. I don't know if any of you have visited. Amazing place, beautiful area, a touristic area, so well worth seeing. My study site is located along the western shore of the Rio Limay, which flows northeast out of the lake on which Bariloche sits. The photo on the right there is a photo of the Limay Valley to give you a little bit of an idea what the habitat looks like. If you squint, you can see a little bit of the river in the background. Again, I'm on the western side of the river. You can see the tents, the little yellow dots. That's my campsite in the foreground of the photo. The tucos are located just on the other side, just this side of the river. And so that kind of helps you orient. We're about a quarter mile from where the tucos are and roll out of the tent in the morning and just amble downhill and go to work. So that's where I've been working ever since 1991 to study this particular species of tuco-tuco. The methods that we use in the field, very simple, very basic. A lot of live capture and marking of animals. We capture just about every individual in this focal population every year. Catching the animals is a story unto itself. I won't say much other than we have to do it by hand. I could talk a lot about our efforts to develop the methodology, but leave it at that. Once we catch an animal, they're all individually marked with a pit tag inserted beneath the skin at the nape of the neck. That's a permanent form of identification. We can follow individuals from year to year. We typically are in the field in the late austral spring, so during the breeding season for these animals, meaning that females are pregnant, late gestation, lactating, pups are still nursing, not yet weaned and independent. As a result, we can track individuals where they've lived since the moment they were born through the rest of their lifetime. We also employ a lot of radio telemetry. Because we can't see a lot of these animals directly above ground, all adults are fitted with a radio collar. You can see the male in the upper right there is sporting a nice yellow radio collar. Once he's released back into his burrow, we can then follow him from above ground and gain a lot of spatial information that allows us to determine who's living with whom, who's sharing a nest site, and really gain a pretty good detailed understanding of social organization. So those are our workforce methodologies, and they've taken us a long way towards understanding the social biology of this species as well as increasingly other species of tuco-tucos. Now, as I mentioned when I started, really the information regarding potential group living in colonial tuco-tucos was pretty anecdotal. So our first step was simply to confirm that these animals are indeed group living. Fortunately, they are. Just to give you some rough indications here, this histogram shows the number of adult females per burrow system for a total of about 85 burrow systems. The data also indicate the gray bars are burrow systems that also did not contain adult males, starting down backwards. Those in red were burrow systems that not only had that number of females, but also had an adult male. The important points to note are the number of adult females per burrow system ranges from 1 to 6, and about half the burrow systems are lacking an adult male at the time we're in the field, again during the breeding season. That seems to simply reflect very high rates of adult male mortality, but it leaves us with kind of a nice comparison in which we can look at the effects of the presence of the male or not on things like female reproductive success. There's never more than one adult male per group, so it's a collection of several females, one adult male. All of those animals share a single communal nest site. So yes, they are communally nesting. And again, important point for what I'm going to say in the next few slides is it's important to note that this means the population in any given year contains a mix of lone females, females that don't have other females in their burrow system, and group living females, females that live in a collection of other adult females. So keep that point in mind as we move forward. But yes, the animals are group living. The second important piece to note about their biology is that all females are reproductive. Again, we're there during the spring breeding season. Every female that we catch is either pregnant or lactating. Now we know from data from lone females, the only female in the burrow system, as well as studies of captive colonial tuco-tucos, that an individual female produces at most about 6 pups per round of reproduction. Yet if we plot the number of pups captured per burrow system as a function of the number of adult females, the graph shown here, you can see that burrow systems that contain more than one adult female are clearly producing more pups than a single female could account for. So yes, all evidence is multiple females, and in fact, all females in a group are reproducing directly. Astute observers of this graph may realize that the number of pups doesn't increase linearly with the number of females, and indeed, per capita, there is a reduction in offspring production as group sizes increase. And that's another story I won't talk about today, but minimally, and again for the critical point here, all females are reproducing, and thus groups of colonial tuco-tucos are plural breeding. How do groups form? It turns out that groups arise due to natal philopatry by females. In contrast, all males disperse. Males disperse at the end of their first summer when they're about 4 to 6 months old, such that when they're breeding for the first time, effectively a year after they were born, they're resident in a non-natal burrow system. They've dispersed, they're not resident in a natal burrow system. In contrast, for females, follow the same pattern. As animals breeding for the first time when they're about a year old, almost 2/3 of females are still resident in their natal burrow system. So there are some females that disperse and are no longer in their natal burrow system, but the majority are still resident in their natal burrow system when they breed for the first time. One result of this is that groups then consist of a series of related females. It's a female kin group with a male who's come in from outside. Males disperse after every breeding season, so it's a different male for each round of reproduction, and thus it really is a female-focused social system. The core is a female kin group. I want to drill down into this just a little bit more because that information has really allowed us to begin to address some of the adaptive bases for group living in colonial tuco-tucos. Here I'm presenting the same information but graphically to try and illustrate the two life history trajectories that female colonial tuco-tucos can follow. They're all born in the late austral spring, and at the end of their first summer, again in that 4 to 6 month range, individual females face a choice, so to speak. Females that disperse from their natal burrow system, follow that top line in this slide, have to find an empty burrow system in which to set up residence. Individuals do not join existing groups. Successful dispersers have to find an empty burrow system in which to live and attempt to breed the following spring. This means that those females that disperse will live and breed alone during their first breeding season as adults. Granted, there may be a male present, they're not that unusual or that weird, but they're not living with other female kin. In contrast, the females that are philopatриc, that remain in their natal burrow system, will live and breed in a kin group during their first round of reproduction when they're 1 year old. Both trajectories, in subsequent years, due to the recruitment of daughters, females following both trajectories will end up living and breeding in a kin group. So the critical difference is that first year, that first round of reproduction as an adult, and whether a female is alone or embedded in a kin group. Again, this comparison has turned out to be extremely useful for exploring some of the causes and consequences of this difference in social environment, whether you live alone or live in a group. Using that contrast for yearling females, for example, we've explored the effects of living alone versus in a kin group on individual reproductive success and survival. Turns out that lone yearling females produce more offspring than group living yearlings, but the probability of the lone females surviving to reproduce in a second season is lower, so potential life history trade-off there. We've also used this comparison to dig into the mechanisms of group living a little bit. For example, looking at glucocorticoid or stress physiology, as well as differences in exploratory behavior, and now beginning to look at differences in neuroendocrine receptor gene expression. Turns out lone females are more stressed, meaning they have higher baseline glucocorticoid levels, and they're less exploratory. The neuroendocrine work is newer. It's suffice to say we're finding differences in receptor gene expression, again in relation to this difference in social environment. So the bottom line is, it looks like, at least for colonial tuco-tucos, the difference between living alone versus living in a group is an important one that arises due to the dispersal decision that a female makes at the end of her first summer. I'll come back to some of this a little later in the talk, but wanted to give you that basic biology for colonial tuco-tucos. The last thing I'll say specifically about kind of in the background of colonial tuco-tucos has to do with the ecology of group formation. That's the question that started all this, that started this comparison. Suffice to say, although, some of the ecological factors that seem to be important to colonial tuco-tucos are the same as those that have been identified from naked mole rats, things like the aridity of the environment, the distribution of food resources. How those things are affecting group living is not the same. What colonial tuco-tucos are doing is not the same as what naked mole rats are doing. For colonial tuco-tucos, to explore the ecology of group living, we've engaged in a series of comparative studies between colonial tuco-tucos and an effectively sympatric solitary species that also occurs in the Lacey Valley, although on the opposite side of the river. So it's really the same habitat, a social species and a solitary species, divided by a significant riverine barrier. I won't go into a lot of detail during the talk today. I can talk more about it later if anyone is interested. But these comparative studies have revealed that what really seems to be associated with group living in colonial tuco-tucos is habitat specialization. For reasons that we're exploring but don't yet fully understand, colonial tuco-tucos are restricted to portions of the habitat, the mesic portions of the habitat. This area of Patagonia is largely arid steppe grassland. punctuated here and there by more music patches. The Argentine term is margin. You can see it on the screen. More music patches that have a different flora, where the soil is much richer in organic materials. And it is clear from our surveys of this region that colonial tuco-tucos occur in those wetter machine areas. They're not out in the rest of the habitat. Again, working comparatively, this suggests that one critical factor, this specialization for wetter habitats, creates a very patchy distribution of suitable habitat for colonial tuco-tucos. The kind of Swiss cheese-looking cartoon over on the right side of the slide, where the dark circles are meant to represent suitable habitat for a colonial tuco-tuco, indicating how patchy and sometimes widely dispersed those areas are, which, again, thinking comparatively, and I'm not providing all the data, but thinking comparatively suggests to us that it's that habitat distribution that really increases the costs of natal dispersal in colonial tuco-tucos and potentially favors filipatry by a fair number of females. That's the very abbreviated version. Don't want to focus on the ecology today, but I did want to return to it because that's the question that started this whole line of inquiry. So bottom line, the ecological explanation for colonial tuco-tucos is not the same as that for African mole rats. And I'm happy to answer questions later or at the end of the talk about that. But instead, what I want to do is, having given you this brief overview of the social behavior, the social organization of colonial tuco-tucos, is return to the larger comparative picture and talk about how comparisons between African mole rats and colonial tuco-tucos, I think, are starting to shape, again, how we might think about sociality, or are emerging as a potentially important comparative system for exploring the reasons for differences in social organization. In particular, I want to highlight one contrast between these two sets of subterranean rodents. Naked mole rats and colonial tuco-tucos are both group living. They both occur in kin groups. There are a number of details that differ between these two systems, but the difference I really want to emphasize today is the difference in breeding structure. It's well known, again, naked mole rats are singular breeders, and I've hopefully just convinced you that colonial tuco-tucos are plural breeders. Is this difference in breeding structure important? Well, increasingly, I'm starting to think that it is. And my first kind of realization that this might be something worth exploring in greater detail came from a series of kind of disparate data sets that have emerged, showing that what plural and singular breeders are doing do not seem to be the same things. This includes a review of dispersal patterns in rodents that I was involved in. It includes looking at the distributions in the brain of certain neuroendocrine receptors associated with sociality, that those patterns differ between singular and plural breeders. It includes evidence from studies of glucocorticoid hormones that how those hormones interface with social behavior differs between singular and plural breeders. And it includes potentially evolutionary evidence, phylogenetically based evidence, that singular and plural breeders have arisen from different origins, different behavioral origins. So kind of a constellation of studies floating around suggesting that singular and plural breeding might be distinct. With that information in hand, I began working with Dustin Rubenstein and Carlos Botero to try and use a review approach to ask, are singular or plural breeding really different? If we could provide metrics drawn from the review of the literature, would those metrics suggest that these really are distinct social systems with distinct statistical, empirical, quantitative distributions? To do this, we ended up compiling data on almost 300 species of group-living vertebrates and invertebrates. The breakdown is shown here. We used data from 88 wasps, 140 ant species, 30 birds, and 35 mammals. It was not trivial to pull this together. We ran into all the typical problems involved in literature reviews. But in the end, the outcome was to demonstrate that indeed it looks as if singular and plural breeding are distinct phenomena. Data for 3 of the 4 of these taxonomic lineages quantitatively, statistically supported that conclusion. And just to give you an idea of how we approached those data, the analyses we were using to address that question, here are data from ants. On the y-axis is the number of breeding females in a group to the total number of females in a group, so really the proportion of breeding females in a group. The x-axis is the total number of females in a group. Both are log transformed as to the differences, the disparities in group size we encountered across this data set. Singular breeders are plotted, the values for singular breeders are plotted with open squares. The values for plural breeders are plotted with closed circles. Because the relationship, that linear relationship shown for plural breeders was statistically significant, we've also shown the 95% confidence interval, the gray area for that relationship. And as you can see, both visually and statistically, it looks like singular and plural breeders fall on different trajectories for this particular metric, again looking at, in a sense, the proportion of breeding females relative to total group size. Well, that was really exciting and really cool, and after all the massaging, the struggling, the working with a large comparative data set, pretty gratifying. As I mentioned, 3 of the 4 lineages we looked at showed a similar result. Now you can almost guess what the exception is going to be. Mammals, of course, the group that I'm most interested in. Things were a bit more challenging there. This graph shows it's the same structure as the one I just showed you for wasps, the same axes. Here you can see, again, the raw points plotted for the different mammal species that we examined. In this case, there was no significant linear relationship for plural breeders, and thus there's no line plotted, no shaded gray confidence interval. Well, that was kind of disappointing. But I think as we've dug into this a little bit more post-publication, there are a couple things I can add that might help convince you that mammals are also playing the same game. First, if we look at these 2 data points down here that stand out, they're marked as plural breeders, but they're kind of right on the singular breeder line. Turns out those are meerkats and African wild dogs. And although by the strict criteria that we applied in our analyses, we had to count them as plural breeders, meaning in some cases there was more than 1 breeding female per group, I think arguably anyone who studied social behavior in mammals, including the researchers who work with these species, would consider them singular breeding. A little bit of a post hoc hedge, but I think that's probably not too far off, which, okay, maybe solves one complication there. That leaves us still with 3 outliers, though, the 3 shown here, which are data from European badgers, the common goombi, a North African rodent, and gray mouse lemurs. And I don't have an explanation. 2 of the 3 of these are not particularly well studied, but I've gone back to the papers multiple times. The data are what they are. Hopefully this will inspire some more studies. I don't have a great explanation. But nevertheless, I think even if we kind of set aside those 3 outliers and squint a little bit, maybe we feel comfortable saying, yeah, mammals aren't completely different from the other 3 cats that we've examined. Nevertheless, collectively, looking across all the taxa we considered, this analysis does seem to suggest or confirm the idea that singular and plural breeding are really quantitatively distinct phenomena. Well, okay, that was a few years out of my life. Why does all of that matter? Well, I think it's important because it might suggest that once again we need to tweak our conceptual framework for comparing different social systems. I'm showing here, again, the continuum perspective, kind of where the story left off the last time I mentioned this. And again, the continuum perspective has not been universally accepted, but it was kind of the second pass in trying to rearrange our understanding of comparative social systems. This new information, combined with all of the various pieces of information that I mentioned a few slides ago, suggests that maybe we need to tweak this yet again. And maybe the scheme should look something more like this, where the critical distinction in terms of thinking about how selection acts, thinking about evolution, should really fall between plural and singular breeding systems. Now, I realize in suggesting this, I probably should rearrange or tweak a whole bunch of other things about this conceptual the way I've laid this out. I agree, but I've left it this way for the moment to emphasize the contrast between starting with the break being everything versus eusociality, to there being no formal break, to now suggesting that the critical evolutionary and selective break is between plural and singular breeding. So, maybe again, these studies of these subterranean rodents are causing us to shift our conceptual framework a little bit. Where do we go from here? What do we do with that? Well, I'm going to suggest, to kind of take us into the last section of the talk here, that we step back a little bit and think about that larger comparative picture. And particularly if we start with naked mole rats and think more broadly about other African mole rats, all members of the family Variability, there are other social and even eusocial representatives of the family Variability. Turns out, taking a phylogenetic perspective, there appear to be two evolutionary origins for sociality and eusociality in this family. All evidence that I've seen, and I would love it if someone knows differently, please speak up and let me know, but all evidence that I've found and everyone I've talked to suggests that all social African mole rats are singular breeders. So they all follow that breeding structure. In contrast, or then the obvious question becomes, what about other tuco-tucos? I've already told you that the colonial tuco-tuco is plural breeding. If we zoom out in the same way for tuco-tucos, what do we find? Well, when I began working with these animals, the expectation, the assumption, was that the vast majority of tuco-tucos are solitary, meaning that each adult lives alone in its own burrow system and has its own distinctive nest site. That's cartooned here on the left, each ellipse representing a different burrow system, each black dot representing a different nest site. But I want to emphasize that was what was assumed. There were no good quantitative data to support that. I then began working with the colonial tuco-tuco, which I've placed at the other end of the spectrum here, whereas I hope I've convinced you multiple adults all absolutely share the same space, the overlapping ellipses, and I didn't present the data that absolutely share one nest site within the burrow system. The question then became, what's going on with the rest of the tuco-tucos, or can we fill in this comparative schema? That was a great excuse for me. I live for the field. That's why I do all of this. That's a big part of why I chose to begin working with tuco-tucos, with the chance to do previously unstudied animals in South America. So great. I was all on board. Over the past decade in particular, have been working to gradually fill in some of that comparative picture. We have, using the same basic methods, live capture, MARP, radiotelemetry, now confirmed that at least 4 species are indeed solitary over on the left side of that scheme. I've already explained the colonial tuco-tuco on the right side, and what's been more interesting and particularly exciting is that we're starting to fill in the middle of the scheme now, encountering species that are social to various degrees, but not as social as the colonial tuco-tuco, meaning they share space, they're definitely using the same burrow systems, but maybe each adult maintains its own private space as well. In some cases with nest sharing, in some cases without. So as you can see, the overall comparative picture for tuco-tucos is expanding. With 60 plus species, it's going to take a while, but at least we're making progress. If we then take a phylogenetic look at this emerging comparative picture, again, it looks like there are probably multiple origins to burrow sharing within the tuco-tucos. This is a partial phylogeny. It's by no means complete. There is no complete phylogeny for the genus yet, although various people are working on portions of it. But this is complete enough to include a number of the key species that I've been working with that are showing different degrees of sociality. And I think taking a quick look at that, may take some squinting in a few moments, but it's a reasonable suggestion that the species that engage in some degree of burrow sharing and sociality likely represent multiple origins of that phenomenon. So some more work to do there, but we'll run with that for the moment. But indeed, I'm jumping ahead here. Again, there are multiple origins to sociality within Ctenomyidae. So, where does that leave us? It leaves us basically with one pattern for African mole rats, one clade, the family Bathyergidae, that exhibits the singular breeding pattern, and a second clade, the genus Ctenomyidae, family Ctenomyidae, that exhibits a plural breeding pattern. Because there are multiple origins of sociality within each of these families, there's some really cool opportunities for comparative research. At the same time, though, we have one lineage doing one thing, one doing the other, so opportunities at that level of comparison to try and explore the mix of evolutionary history and current ecology that has led these lineages to follow two different breeding trajectories. So just in the final few moments here, to kind of bring this back or tie this into, again, where my research program is headed and what I'm thinking, we move to the wildly speculative part of the talk. Everybody has to have the speculative conclusions, right? Here come mine. And where my thinking is going at this point. Multiple researchers have linked singular breeding in particular to monogamy. And indeed, it's been argued that singular breeding arises evolutionarily from monogamous systems. Multiple papers have been published suggesting this relationship, and again, in some cases, exploring it phylogenetically. So, an evolutionary link there worth pursuing. At the same time, on a more ecological scale, the classic factors thought to favor monogamy are either the need for male parental care and or limited availability of females, meaning males really don't gain much or gain less by leaving a female, trying to find other females, compared to staying with a female and maintaining future reproductive opportunities with her. So well-established ideas, both evolutionarily and ecologically, that tend to link monogamy to singular breeding. Can we start to apply any of this to these two sets of subterranean rodents, the mole rats and the colonial tuco-tucos? Well, to me, where my thinking keeps going is back to patterns of dispersal and how colonies in both sets of rodents form. For colonial tuco-tucos, I already mentioned, animals that disperse have to find an empty burrow system. Females don't join existing groups. Males do, but again, focusing on females really have to find an empty burrow system in which to live to be a successful disperser. In colonial tuco-tucos, new colonies form when a dispersing female meets up with a dispersing male, and they occupy a burrow system together. So as a male-female pair, or if the male doesn't make it, a lone female during that first breeding season. Absolute pattern in colonial tuco-tucos. Somewhat surprisingly, the same pattern seems to hold in African mole rats, both naked mole rats, and now I'm going to invoke the second social species, the Damaraland mole rat, which is a part of that other origin of sociality. But based on the data I've been able to glean from the literature, as well as talking with colleagues who work with naked mole rats, it again appears that most dispersal events, most new colony formation events in these animals also result in a single female meets up with a single male, and they establish a new colony together. So, for that much of the story, tuco-tucos and mole rats seem to be doing the same thing. New colonies form following the same pattern. It's after that things start to diverge. In colonial tuco-tucos, as I've already mentioned, males disperse at the end of every breeding season. So that new partnership is at most only going to last one breeding season, after which the male is going to leave. The female may recruit daughters to live with her, but it's a new male who will move in for the second breeding season. Sets up a polygynous system, definitely not monogamy, or at least not monogamy across seasons. In contrast, all available information that I've been able to get, and again I'd welcome if anyone knows differently, is that in the mole rats, particularly in naked mole rats, that pair that founded the colony is going to stay together across multiple seasons, across multiple years, as effectively a monogamous pair. So somehow there's something that even though colonies start out in the same way in both lineages, something's leading to very different outcomes in terms of patterns of subsequent dispersal and patterns of breeding structure, meaning the potential for polygyny versus monogamy. Well, this leads me back to thinking then about, maybe this would be a convenient point in the colony cycle of both sets of animals to start to think about those classic factors favoring monogamy, such as need for male care or the availability of other females. Speaking for the data for tuco-tucos, which I know best, male parental care is not needed. A female can rear her pups just fine on her own, and there's no difference in the number of pups she will produce based on whether a male is present or not. Male care doesn't matter in colonial tuco-tucos. This is more difficult to quantify, but just given the distribution of colonies within a habitat patch, also seems like, relatively speaking, it's pretty easy for a male colonial tuco-tuco to disperse and find another set of females. Distances between colonies are often tens or at most hundreds of meters, and clearly males are shuttling around with no problem between breeding seasons. And then finally, the last piece of information that I'll add is in colonial tuco-tucos, about 25% of newly formed colonies or groups persist to at least a second year. The question now is, how do those things compare in the African mole rats? I don't have the data. I've been working to try and gather some of it from the literature. I would welcome input or collaboration with any of you out there working with the mole rats. So instead, I'll phrase these as predictions. I would predict that in a newly formed colony, male parental care is much more important in African mole rats. I'll also predict that in the African mole rats, finding other females is more challenging. That may also favor males staying with a given female. And finally, and this is probably the easiest one to actually quantify, is I'll predict that relative to colonial tuco-tucos, the persistence of new colonies of African mole rats is much lower. To date, the only number, or not even a number, statement I've been able to find is that persistence of new colonies of naked mole rats is low. But again, I would welcome the opportunity to work with any of you who are studying these animals to try and fill in some of these gaps. So I'll leave you with those wild speculations, as we're kind of obligated to do at the end of the talk. But that's where my thinking is going right now, as to what might be a tractable route to try and start exploring in an ecological time frame why reproductive patterns in tuco-tucos and mole rats may differ. Just to wrap up then, what does it all mean? Hopefully one thing I've managed to convince you of today is that tuco-tucos are emerging as an intriguing new study system. What they're doing is more variable than anyone expected, and I think is creating some interesting opportunities for comparative studies within this family of rodents. Certainly it's keeping me going. I can't wait to get back to the field and continue studying new species and filling in that comparative framework. At the same time, I hope I've convinced you that comparative studies of different subterranean lineages offer some important and I think growing opportunities to examine the factors shaping different social systems. And even if my speculations about new colony formation and what's going on in tuco-tucos versus mole rats are wrong, that those types of comparisons, as we gain more information about all these rodents, those types of comparisons, digging in like that, may be more tractable and may end up being informative. But for today, I'll leave it at that and just say thank you for listening, and I'm happy to answer questions. Thank you.
Great. Thanks so much, Eileen. It's a really great talk. So we do already have some questions from the YouTube chat. So to start, Saeed asks, what would be your suggestions and further questions for next steps of the study? I know you discussed a lot of studies, so maybe if you want to choose one.
Yeah, because I guess my first question would be what part of the study, the tuco-tuco specific part or the comparative? I don't know if Saeed can respond or if I should just pick one and go with it.
Pick your favorite, and then I'll let you know if he says anything in the chat.
Okay. With regard to the tuco-tucos, and actually going back to the colonial tuco-tucos, the one I know best, where work with them is going right now is looking in much more detail detail at what determines whether a female disperses or not. But this is kind of the last big pulse in my career before I retire. But I showed you data from the field indicating that some females disperse, some don't. That difference occurs even among littermate sisters. So from the field, we have information about kind of the setting, meaning, group size, the density in the burrow system, something about the ecological conditions. I also have, and have had for 25 years now, a captive colony of colonial tuco-tucos in Berkeley that are upstairs a couple floors, that is allowing us now to look at that same question, but in a much more mechanistic approach, that we breed lots of tuco-tuco pups every spring. We can, in a sense, profile them, run them through a series of standard behavioral tests to ask who's more exploratory, who's more tolerant of unfamiliar conspecifics. We've created kind of a dispersal assay, so who's first to leave the burrow system. Pull all of that together with things like body weight, glucocorticoid levels, and we're really starting to develop, I don't want to say too much yet, but really starting to see a dispersal profile. It's not like it's been suggested for naked mole rats where there are necessarily pronounced morphological differences or dispersal more, but more on the level of individual variation, starting to get closer to identifying what determines if this female is going to disperse and follow that trajectory versus stay home and follow the philopathic trajectory. I have no idea if that's what Saeed was thinking, but I ran with it, and that's a big next step.
Yeah, so many different ways you could take it. Okay, and then so Chris also has a question, and he made a correction after the question, so I'll insert his correction. Okay. Do you know if the dispersing singular breeding living female tuco-tucos, so C. sociabilis, are neurologically different? So i.e. possibly less socially tolerant, different oxytocin receptor patterns, etc.
Which I kind of just touched on, right? We don't have all the answers yet. I'm working in collaboration with a new colleague here, Annalise, to answer those questions. That we've now completed the first round of doing all the behavioral profiling of a cohort, a generation of female pups, looking at glucocorticoid levels, and then we did end up sacrificing a subset of those females. We have the brains, we just haven't gotten around to starting to work with them to look at either receptor distribution, receptor expression, things like that, but it's in the plan, yeah.
It's upcoming. And then Ebby says, fantastic talk. Are there hard limits to how many females can live together? So with more sampling, would you find groups of larger than 6 females?
Excellent question. I mean, I don't mean this to sound like a smart ass answer, but are there any hard limits in ecology? That said, realistically, yes, there probably are upper limits in terms of, as with any group living animal, there are benefits and there are costs, right? I mentioned that there does seem to be a per capita reproductive cost to being in a group. I'm sure there are parasite costs. These things are loaded with ectoparasites at least. We have the data, we haven't looked it up, but presumably that's more of a cost in a large group. There's also simply how far you can expand a burrow system, since you have to do that to acquire food resources. So it's probably a complex mix, but my guess would be there is eventually an upper limit in terms of that cost-benefit ratio. Yeah.
Great, thank you. And then, so that's it for the YouTube chat questions. Everyone else, like Saeed and Gabrielle, are just saying, great talk, thank you. I did have a question of my own, actually. It was kind of near the beginning of your talk, and I don't know if you covered it later. So you had said that lone females usually they're more stressed, they have different, cortisol receptor distribution, they're less exploratory. So that's sort of isolation or lone living leading to those behaviors. Do you think there's any behaviors that lead to lone living? Like, are females that live alone more aggressive to start off?
Yeah, which kind of gets us back to the stuff going on in the lab right now, trying to figure out what dictates these dispersal decisions. Again, very preliminary, but the connections we're starting to see, this is the part, this video will self-destruct in 2 weeks, which is good, right? within a litter. So again, we're looking within a litter-mate sisters. It's the larger female or females in the litter who are more exploratory, higher baseline GC rates, less tolerant of an unfamiliar conspecific, and in this dispersal assay tend to be the first to complete the task. So to the extent that that at all imitates dispersal, that starts to be emerging as kind of a suite of factors going on. Also, very anecdotal, but some suggestion when we leave these families together in the lab the way they would live in nature often, and if aggression is rare, but if there is, it seems to be between the mom and the largest daughter. So kind of starting to plant the idea that we don't see any overt dominance hierarchy, we don't see any overt reproductive suppression, but that maybe there's something a little more subtle, and it's body size is part of that. Is that maybe starting to touch on your question, Amanda? You guys, all is super, this is great. It's justifying why we're doing this latest pulse of stuff, but we don't have the answers yet. Yeah.
All to be determined.
Yes, yeah.
Great. Thank you so much. So that is it for the questions. So thank you again, Dr. Lacey, for a great subterranean rodent talk. We learned all about the distinctions between the naked mole rats and the other subterranean rodents. And also thank you for everyone who tuned in to watch. So this will be available for 2 weeks if you want to watch after the fact. And our next seminar will be taking place on June 28th from 2 to 3 p.m. GMT. So we hope to see you all there. And thanks again, everyone. See you next time.